The workflow presented here can help extend our understanding of the cellular role of chaperones during oxidative stress conditions. Specifically, it can help to understand how do chaperones prevent protein aggregation during unfolding conditions.
Though these two buckets of different techniques provide insight into the activity of Redox-regulated chaperones, it can also be used to study other protein-protein interactions, such as enzyme-substrate interactions and protein dynamics.
Visual demonstration of this method is critical as it involves the usage of different instruments, which may be challenging to new users.
First, spin down a previously thawed protein sample to remove aggregates. Then add 5 millimolar DTT and 20 micromolar zinc chloride and incubate the sample for at least 1.5 hours at 37 degrees Celsius. To remove the DTT, equilibrate a desalting column with a 40 millimolar potassium phosphate buffer by filling the column completely with the buffer and letting it drip out. Using tweezers, gently push down on the white disc filter in the column and remove it. Refill the column with potassium phosphate buffer and centrifuge at 1000 times G for 3 minutes. Following centrifugation, transfer the column into a clean tube and add the protein sample slowly to the middle of the column. Centrifuge the column at 1000 times G for 2 minutes so that the DTT-free protein is in the flow-through. Now check the protein concentration and measure its absorbance at 280 nanometers. Distribute half of the protein samples into aliquots. Incubate.